All right, guys, so we are going to go through our protein synthesis worksheet here. You should have made a copy. I'm going to give you a few minutes to just kind of pause this video and take the time to use dictionary.com to find the definitions for these. When you are looking at transcription and translation, make sure that you are looking at the genetics definition. There's a bunch of different definitions, and it'll have it like in parentheses what topic that it's for. So you can go ahead and pause this video and then go through those definitions. And then unpause, and we will kind of check those and continue on with labeling our diagram. All right, so you should have your definitions. We're going to go through them here. So messenger RNA is what copies the coded message from DNA. So when that DNA unzips, it's creating that new messenger RNA strand, and then it carries that message to the ribosomes. Then transfer RNA carries amino acids to the ribosomes and adds them to the growing proteins. The transfer RNA reads that coded message so that it knows which amino acid to add, and it builds that protein molecule. Transcription then is the first step in well, it's one of the first processes in protein synthesis, and this is where that genetic information from the DNA is synthesized, or it's like copied or duplicated into that RNA strand. Then translation is the second like big overall step of protein synthesis, and this is when the mRNA specifies what sequence or like what order the amino acids will be put into and how they'll build upon that protein. Then two other terms I wanted you to define, because we'll hear them in and we'll see them in our labeling diagram, and then some other activities that we're going to do later on this week is codon and anticodon. So a codon is the triplet of nucleotides, which are those DNA bases or those RNA bases. So the codon is on that mRNA strand, and then that gives a specific sequence or a specific code to determine what type of amino acid is used. Then the anticodon are three bases that actually like hang down from like the RNA or the transfer RNA as it's reading that message, and it reads that coded message, it matches up with the right codons, and then it adds the correct protein. Okay, so once you have these definitions filled out, we are going to work through labeling our diagram together. As you can see, you have the protein synthesis diagram, and we're also going to watch a couple videos. So before we get started with our diagram, I did kind of want to show you just briefly the difference, the two main steps of protein synthesis. So the first one we're going to watch is about transcription and how that happens. All right, so that was transcription. So pretty neat, pretty interesting, showing how that DNA strand unzips. Those free-floating nucleotide bases come into that factory and are able to read and translate that message and create that mRNA strand. So if you click on this diagram, at the bottom of the picture, you are going to see the edit button. So we're going to click on that, and we're actually going to go through and fill out this diagram together. So at the top here, that double helix strand, you guys should know or recognize that as DNA. Then the video we just watched talked about that first step of protein synthesis. That is transcription. Okay, so it's reading that information from the DNA and creating that mRNA strand. So as it unzips and creates that strand, this is going to be mRNA. Now, this is all currently occurring in the nucleus. That's where DNA is. That's where that mRNA strand is created before any major protein synthesizing occurs. So we are going to hold on to this, and then we're going to watch a video about the next step of protein synthesis. So this step is now translation on what happens to that mRNA strand and how the protein is created. All right, so that went through the next process and combined a few more of those components. So the second step of that protein synthesis is what we call translation. That is where that information is now being read, that coded message is being read to know what amino acids to use to be able to create that protein. So this location, if you remember at the beginning of that video, the mRNA strand leaves the nucleus and now enters the cytoplasm of the cell. So we still have our mRNA strand here, so that big strand that looks like a ribbon. We're going to go over to the right here. That is still our mRNA strand. The section that is in this little ribosome, that purple circle here, that is what we call ribosomal RNA or rRNA. It's just a fancy term for recognizing that that section of the mRNA is in the ribosome. Then what we have is our other type of RNA, which is that transfer RNA, which carries those specific bases and those amino acids. So this funky little shape, the molecule is our transfer RNA or tRNA. And at the top of that, that little circular dot is what represents the amino acid that's at the top, that as it reads that coded message, it will add that to the chain that it's creating for the protein. Then on the transfer RNA is where you have your anticodon bases. So there's 3 bases that will read the coded message or the codon on the mRNA strand and be able to correctly identify which amino acids used. And as you can see that process, the amino acids then create this growing chain, which is the growing protein. Now, like the video showed, that protein is going to continue to grow until it reaches a stop code and it's completed. And then it will be released and it will go where it needs to go. Once you have this chart filled out, please make sure you hit save and close. And then you guys now have the protein synthesis diagram filled out. Please make sure that you submit this so you have it as a reference. Otherwise, this is our worksheet and you guys can finish up and exit out of it.